There is a link on the chat with the attendance register, so you're welcome to complete those details in the meantime. Very good evening, colleagues. The time is 6:30. A very warm welcome to another session of our Intellectus Campus Wednesday evening sessions. My name is Obakeng Mwatse. I serve as the General Manager of the Intellectus Campus Group, a skills development and training institute that is currently having 8 campuses across 5 countries, namely South Africa, Botswana, Namibia, Zimbabwe, and Eswatini, soon to be opening up a campus in Zambia. The session this evening is being delivered by Dr. Martin Wucher and will be moderated by our very own Dr. Jacqueline Stone, who will run the introduction and bio for Dr. Wucher. Before we dive into that, just a couple of housekeeping reminders. Colleagues, this is intended to be an interactive session. In other words, your participation is very much welcomed. In order for you to participate, you can apply one of 2 ways. Either raise your hand and a member of the Intellectus team or Dr. Jackie Stone will recognize you for your contribution. Alternatively, please make a note in the chat, and that will be read out if you're not called upon. In the interim, we are, however, requested to please remain muted for the duration of the session, and you will unmute when recognized. Please complete your attendance details on the link that is being shared on the chat. This will enable us to circulate your certificate at the end of the session. If you do not complete your details per the link, we may inadvertently overlook sharing your details. So please do not post your details on the chat. Please opt to use the link. A very warm welcome to yourselves, trusting that you will enjoy the session. Good evening, Dr. Wucher. A very good evening, Dr. Jackie. Please proceed.
Thanks very much, Obi. Welcome everybody to this evening's session. I'd like to introduce Dr. Martin Wucher, who is associated with the Namibian Dental Association and holds an associate membership with the South African Dental Association. He earned a BSc degree in zoology and microbiology from Rhodes University in South Africa and transitioned into the field of dentistry, where he obtained a BCHD degree from the University of Pretoria and approved an approbation. I hope I said that correctly because this is in German. Als Zahnarzt, licensed to practice as a dentist in Germany. I probably pronounced that dreadfully, but never mind. You can correct me a bit later. Where he actively participated in prosthodontic practice. Additionally, he received a postgraduate diploma in periodontology from the University of Pretoria, followed by another postgrad diploma in aesthetic dentistry and a master's in dental science from the University of Stellenbosch. Martin is officially registered with the Health Professions Council of Namibia and has extensive experience lecturing on various subjects within the realm of dentistry, as well as human and business management. His academic contributions extend to his role as a part-time lecturer and co-chair at Dresden International University, where he contributes to the postgraduate masters in Preventive and Functional Medicine. So welcome, Martin. That is an interest of the group is functional and integrative medicine. And Martin's specific focus is on chronic diseases, mitochondrial medicine, and frequency medicine. So the floor is yours, Martin. We look forward to the first of about 4 talks that you're going to give us, reminding us that our undergraduate learnings were important.
Good evening, everyone. It's a pleasure to be with you, and I send greetings to my esteemed colleagues on the Southern African continent. So welcome everybody tonight. What I'm going to do tonight is give you a rundown, a perspective of some of the, I believe, most important underlying events that happen in our body at cellular level when we move our body from health into chronic disease. So I've taken the topics as up in the front of you, and we will focus on especially the mitochondria because they are key to this whole game. It'll be a lecture that's got quite a bit of detail, but don't panic if you don't follow all the time. It doesn't matter. You only have to come away today and take a big message, and that I will explain at the end of the day, end of the lecture. So without further ado, I'm going to share my screen. And I think we can just delve into that. Okay. Can you guys all see that? Is that visible?
It's perfect. Thanks.
Okay. As you have heard, I live in Namibia. I'm a born and bred Namibian, or bred and born Namibian. My family's been here nearly 120 years. So you could say my heart and my soul is anchored solidly in our continent. And I'm prepared to die here. And I'm very patriotic about our continent. I still think we live in one of the most beautiful places on earth, and we've got endless opportunities if only we manage to look at them. So let's start a little bit near Windhoek. I'm just 60 kilometers north of Windhoek, which is in the middle of Namibia. You all know where Namibia is. And as Jackie mentioned, I started off as a young guy, and remember, when you're young, you use 80% muscle and a little bit of your brain, and then when you get to my age, your muscle isn't what it used to be, so unfortunately you can't use it as much, but then you have got the advantage of using experience, meaning your brain. So without further ado, let's delve into something that I have observed. Because of my biology background, which included ecology, how animals work together,
Next speaker.
how everything works in nature.
Okay.
Pride, can we make sure everyone's
Microphone
muted,
is-
please? Please, everybody mute your microphones.
Namibia is an interesting place. It has very high level up-end Namibians that live in effluent environments. And then we've got the most rural populations like we all have in our different countries. The one is, you could say, living in a westernized lifestyle, and the others, virtually, as you will see in this photo, have not even got any shoes on them. They own very little as in terms of financial issues. Sorry. But there's one thing that's really interesting. The westernized middle class, normally when they get to middle age, they get older and older with more and more medications. And then when you get to the rural areas where people have got very little, you hardly find any, you could say, civilized or modern chronic disease. You will hardly find blood pressure issues. You will not find cholesterol issues. You will not find diabetes. The people look healthy. The first thing you notice is that there are no fat people. There are no fat people that have got metabolic syndrome. And the moment these rural folks move into cities and start working at the local service station and start drinking the cool drinks and eating the chips and the fast food that is available there, they end up in the same situation. They gain weight, they acquire all these symptoms, and they all end up in the same way that these more affluent groups are at the moment. So today, we're going to talk about chronic disease and the tip of the iceberg, which is cancer. So I'm going to use cancer as an example. And cancer is, let me just share something here. Okay. All right. Cancer is a good example for other chronic disease because it's really the tip of the iceberg, the stuff that is very visible in the end. Chronic disease are the issues listed below. This slide is from, I think it's 2019 or something, but very little has changed. In fact, if any, things have got worse. If you look at this one, and this is by the WHO, also from that year, it's an incredible rise in chronic disease. And I think with the event of COVID and the aftermath of COVID, these statistics are at least that, if not more. So this is just as a background. If we look at cancer, it's much the same story. Cancer is slowly growing, and we just take it as a modern phenomenon. I think very little is, we spend very little time looking at the ecology of it. Why is it happening? What are the driving factors? And we hear the normal stuff. In other words, lifestyle, maybe smoking, unhealthy lifestyles, toxins in our environment, and that sort of stuff. And you can see number one is cardiovascular, and then cancer is number two, and then all sorts of other ailments and diseases that follow that. So if we look at cancer, and this is 2018, and I try to find newer updated numbers. They're not available at the moment, and I'm sure they will be higher than expected, because as we know, in the COVID area, especially in those countries that have a lot of vaccinations, cancer and turbo cancers, as they call them, are popping up like crazy, like mushrooms in a dung heap. So cancer statistics, once again, this is new stuff, and we can see the most predominant is prostate cancer in men, and then of course breast cancer in women, followed by lung cancer and colon and urinary tract cancers and that sort of stuff. This is just a background. Why I want to show you these graphs is basically to highlight the fact that if we look from 1975 to now, and medicine has supposedly advanced such a lot, you don't see a critical fall in these diseases. In fact, they are much the same. And remember, they are per 100,000 people. So the relationship should be the same. If anything, if you look here at 1975 and you look at now, which is basically 50 years or close to 50 years, lung or prostate cancer is pretty much where it was then. In fact, it's quite a bit higher. It's gone from 100 to say 120, and the same with breast cancer in women. Some cancers have dropped. This is lung and bronchus in men, and I think it's been described as because the smoking cessation of men, but at the same time, women have started smoking, so their cancers have gone up. All right, so why is this cancer growing? As I've said, is it just bad luck, or is it just part of the earth? Have we changed? Is it genetic differences? Whatever it is. If we look at the background and the reason for cancer, this is what we find. The National Cancer Institute in America basically explains that cancer is a disease of genetic change, of mutation. The cells have got genetic damage and they do whatever they do. So let's look at a bit of the background research and then come to a new model or a different model that I want to show you and present to you today. So a cancer explanation presumes that here on the left there is a cell that is healthy. This is supposed to be the cell. This is the nucleus. So there you have got one mutation in these are supposedly the genes. And the cell still looks pretty okay. Now you've got two mutations, and you can see there is a little bit of change happening. There are more mutations and then more. And this is when the cancer really takes off. So this is what we call the somatic mutation theory. And we've got it in a different graph that we can say carcinogens damage the DNA, and that leads to abnormal growth. And once that growth has taken off and is not controlled anymore, we get different sites throughout the body, and then we call it metastasis. So, but then the guys started asking, and they said, okay, say you have one mutation, two mutations, say you've got 100 mutations in a cell, and that cell then forms a whole cluster or a tumor of these cells. Then if it's due to the genetic aberration, there should be a similarity in those genetic aberrations. Now, strangely enough, that does not happen. And sorry, Jackie, can you hear this screaming at the back?
No, I can't.
Can you hear my voice clear?
Yeah, we can hear your voice beautifully.
Okay. So here we are. If it was a genetic mutation, or say 100 genetic mutations that led to that bunch of cells going haywire, they all should have the same genetic differences. But when you look at a tumor, there are several million. There are millions and millions of differences. So how can that be? There is no good answer for that. Then in about 2000, two very well-known scientists, Hannan and Weinberg, came along and they published this article in Cell. And they said, look, there are 6 things that we can see in cancer cells. First of all, they grow like crazy. They just divide. As long as they get nutrients, as long as they're in the right environment, they will divide. Secondly, you will see vessel formation or capillary formation, angiogenesis. In other words, new vessels will grow into that tissue and supply it with needed nutrients and food, and the cancer will take off. Also, these cells are not bound by intercellular communication anymore. It's just like a bunch of people that say, listen, I'm not following the municipal rules anymore. I will take any piece of land and I will just put my shack on it and I will grow and I'll do what I will do. They also lose to a large extent, the facility of apoptosis. In other words, if a cell is damaged, if it's not doing the right thing, there are certain triggers and switches that can be switched on and the cell will die you could say a processed cell death. It doesn't just explode, it gets dismantled in a fashion, in a specific fashion, and the components or some of the components are used by other cells. Number 5, the cell does not die. So if you look at something, HeLa cells are used in science and they have been used for the last 70 years. They came from a metastatic cancer. I think it was a liver cancer of a specific person. And these cells just carry on growing. They cannot age. And then the last thing that we see is metastasis. In other words, the cancer leaves the site that it's in and it moves to all other body parts. Then that was accepted and most people said, well, that's cool. It makes a lot of sense and this gives us a good description of cancer growth. Then about 12 years later, or no, 10 years later, roughly 10 or 11 years later, they added another 4 categories. And they found that cancer cells have got the capacity to circumvent or suppress our immune system. It also promotes inflammation, and those 2 have a synergistic effect. In other words, it causes inflammation, which in case then suppresses the immune system, and that helps the cancer environment and the cancer growth. Then also there's genetic instability that has been said all the time, and mutations, we can see that. And number 4, and I'll put this in bold, there are changes in cellular energetics, and this is a really important factor. So this is old news. This is what we would call the dogma. This is what everybody's been saying. So what is the big story? Why do children, when they are very young, get cancer? Now, if mutations have to pile up to get to a certain level before the cancer starts becoming a cancer, then little children should have a much better chance. And we should hardly see any cancer in young kids because they just did not have the time to accumulate those mutations to form a cancer tumor. Why is every tumor different? This is a question that we've had before. They should all have the same genetics. And then, not only that, every cell, individual cell, is also genetically different. So this is really, really strange. Remember, we said cancer is a genetic disease. And at the end of the day, after Nixon declared the war on cancer, I think in 68 or 70, around there. So after 50 plus years, we still have got no 100% and really good answer. We still have got 3 tools only to fight cancer. Number 1, surgery. Number 2 is radiation. And number 3 is chemotherapy. And we all know that they are pretty destructive to the patient. This is an interesting picture, and I've taken it on purpose. I think we all know when we stay in our houses in Africa, we've got lots of insects and we've got these flies. Eventually, they end up on the windowsill just below here and they die. Meanwhile, the window next door is open. So what I'm trying to say is there must be another way, and we must just find that other way. So how did we get to another model? Dr. Thomas Seyfried, he's a biologist, PhD biologist. He's been at Boston College or Boston University for the last 40 years. He's virtually spent a lifetime researching cancer cells and the metabolism of cancer. He's worked with many, many different groups and scientific teams around the world. And he came along and he said, look, there must be some reason here. They went along and they looked at stem cells. Now, stem cells are undifferentiated. They have got the basic cell structure and they've got the nucleus. And with modern techniques, you can insert a small little needle. Normally, these needles are made out of glass because it's not that detrimental. It gets pushed through the double lipid membrane, and you can suck up and manipulate certain organelles or even the nucleus. And this is what it gets used in this type of research. So here we are. Here's our normal cell. We've got the nucleus up here, the endoplasmic reticulum, the rough one and the smooth one. We've got a Golgi body, we've got fibrils, and we've got our big friends, the mitochondria. And we take this cell and we take it from a frog and we start doing research on it. So the first test they did, and it was McKinnon et al., and they took a cancerous cell, assuming that the nucleus had mutations that caused the cancer. So remember, the argument is the cancer is inside the nucleus because everything is mutated. And now they put that nucleus in a normal cell here on the left. And they let it grow. And out of it comes a normal tadpole. And that should never happen because a cancer cell, which comes from a lung kidney tumor, should actually grow a lung kidney or a lung, a kidney tumor, frog kidney tumor. It should grow another kidney tumor. But it grew a tadpole, which is a healthy cell, a healthy basis. The tadpoles didn't make it till frog. They died early on, but the basic thing was the same. So then they did the same with mouse melanoma cells. They took the supposedly diseased nucleus, put it in and removed the nucleus of this cell and put the cancer nucleus in here. And guess what? The mouse embryo grew normally. Eventually it died. So that is really strange. How can that be and what's happening here? So this cannot happen if we believe the story that the nucleus has got genetic mutations and they cause the cancer growth. There must be something else. So then came Siegfried, and he did these experiments in 2014, you could say virtually 10 years ago. And that was interesting. He took a cancer cell, and of course, the cancer cell produces, now this is a normal cell. The nucleus is the nucleus here, and you see these other green dots that are put in there are the mitochondria. They are all normal. Then you've got a cancer cell, and everything is red. And of course, as we expect, everything becomes red. One cell grows another tumor there. So then they did something else. They took this cancer nucleus, supposedly again, and put it in a healthy cell. Remember, they removed this nucleus and put the red one in. And guess what? Out of that grew a normal cell. And not, as we expected, a cancer cell. Then they did it differently. They took a cancer cell, like this one. They removed the nucleus and put a new nucleus in. And guess what? It became a tumor cell. So what I'm trying to say here is, if you look at this carefully and you think back and forth, every time we changed the nucleus, nothing happened. But here, we changed the mitochondria. Can you see that? And the mitochondria influenced the tumor growth. So we had a healthy nucleus with sick mitochondria, and it caused a tumor. So from that, they said non-cancerous mitochondria can inhibit tumor properties. Otherwise, other way to say it, if the mitochondria are sick, you will get cancer. And if you've got healthy mitochondria, you will not get cancer. So what did they do here? They took tumor cells, removed the mitochondria that were sick. The red ones are sick mitochondria. and they put new healthy mitochondria in it. And guess what? The cells became normal. This is crazy stuff. Because, and then also the offspring were normal. So the cancer growing mechanisms were removed or suppressed by adding healthy mitochondria. And this is a huge, huge change in thinking. So then Seyfried went along and he says, okay, what do mitochondria have to do with this whole lot? And they looked at many different cancers, and they specifically looked at the mitochondria. And the mitochondria inside the cytoplasm, they did not find any normal mitochondria in the cancer cells. There were many, this means changed mitochondrial structures. So they had all different things that were done or that happened to be different. Human tumors had changes in their mitochondrial DNA. Okay, and we'll talk about that a lot. And this is really important. The oxidative phosphorylation in these mitochondria was all damaged. It didn't work properly anymore. So Thomas Seyfried then made this statement. He says, it's my opinion that chronic respiratory insufficiency caused by continuous environmental triggers constitute the main cause of cancer. In other words, something in our environment damages the mitochondria. They cannot really do their oxidative phosphorylation anymore, and then the cell becomes cancerous. This is something very similar to what Otto Warburg said in 1965 or he only worked in 1962, but anyway, in the 50s, in the 1950s. He was a famous German scientist. He also got a Nobel Prize for, I think, biophysics or something. And he focused a lot on this research on mitochondria. So now we've spoken about mitochondria. Let's have a look where they come from. In ancient times, the earth was very different to what it is today. The oxygen levels were around 4%. There was a lot of sulfur. There was a lot of methane. There was a lot of nitrogen and other stuff. And bacteria and these little organelles that grew at that time couldn't really handle a lot of oxygen. And when the plants started growing and the algae started growing, the atmosphere changed drastically because those green algae started picking up CO2 and making oxygen or giving off oxygen. And then these bacteria that could not use oxygen were under stress. So what did they do? They used some organelles and fused these together and they formed the protista. In other words, it was a cell that had the archaea as a mitochondria inside them, and they formed the forefathers of every multicellular living organism on this planet. So they are the plants, they are the fungi, and then somewhere here are the insects and everything that lives, and here we are, the animals, where the vertebrates and the mammals, and then obviously the hominids, the humans are in this bunch here in this top. So long story short, bacteria that could use oxygen and cells that could not use oxygen fused together, and they formed this symbiosis, and this is what we have. We have the cell, and these little guys could use oxygen, and they went inside, and this one protected them, and they helped this one do their thing. So this is the mitochondrium. This is called the outer membrane. This is the inner membrane. It's folded up and down. It's got cristae, and this jelly-like part in the middle is called the matrix, and the space between here is called the intermembrane space, and we'll talk about it quite a lot. So as humans or as mammals or as animals that mate, where do the mitochondria come from? They come from the mother. They are passed down in the ovum, and they stay in the ovum, and they grow with the ovum. On the father side, the mitochondria are virtually all in the tail end of the sperm. And the moment the sperm head moves and dissolves this layer on the outside of the ovum, the tail gets separated. And with that, all the father's mitochondria are lost. So the crucial thing is that the mothers are predominantly, I'm saying 99% responsible for the mitochondrial genetics and the mitochondria that the offspring will have. So the mother will give them further, and much of the mitochondrial DNA is from the mother. And this is used in the research of human evolution. And we all have heard the story of the the mitochondrial Eve, or the mother, the genetic Eve, the mother of everyone that came from Africa, and they used this type of research. They looked at the mitochondrial DNA from the mother. Okay, inside the mitochondria, we have got little loops. Now, that's also interesting. In humans, the DNA is in long strings packaged into chromosomes. In mitochondria, it looks much like a plasmid. It's a little loop. And it fills a lot of the cell, up to 50% of the inside of this, or actually the mitochondria fill a lot. In many cells, we can get up to 20,000, like in your heart cells, in your cardiocytes, in the brain, we've got a lot. And then we have got these mitochondria that are in long strings or single. And we've got these mitochondrial DNA loops. The mitochondria only have got 24 genes, and they make 14 proteins, polypeptides. And the remaining genes come from the cell, from the big cell. You could think the mitochondria are like algae in a dam. They can grow into long strings, and they can fuse together. In other words, if two healthy mitochondria get next to each other, they can fuse. And this is a really interesting thing, because they can also, you can see these dark little places. If there are some genetic fragments that are not healthy anymore, they will all be pushed to one side. You can see it here at the top. They are all pushed to this side, and then the mitochondrion splits, and the side with the rubbish in gets eaten up by mitophagy. So we will talk about mitophagy later, but the mitochondria in this way have a way of sorting out the ones that don't work properly anymore, and they can put them on the rubbish bin and they get dismantled. It's like a chop shop for old cars. Okay, so they push them away. These are cardiocytes. So this is a heart muscle cell, and you can see all the mitochondria sitting here. This is the nucleus of the cell, and you can see them here in the cytoplasm. At bigger magnification, we can see here, this is the intermembrane space between the two, and these are the cristae, the folds that go into the inside. And this is the outer membrane. And in the middle here is what I said to you is a matrix, and inside the matrix, we have got the DNA little rings. Okay, in the intermembrane space, now this is the cell, this is the matrix at the bottom, and this is the intermembrane space. And on the inner membrane, on this membrane, we have got what we call the electron transfer chain. So I'm not going to go into detail, but a certain amount of nutrient or fuel, and in this case acetyl-CoA, is pushed into complex 1. Electrons are exchanged, and it's moved down the road. And in that process, complex 1 and complex 3 are proton pumps. And please remember that word, proton pumps. They pump protons from the matrix into the intermembrane space. So eventually you'll have lots of protons in the inside and very few in the matrix. And then these protons will get pushed back through complex 5. It's called ATPase. Okay. And the ATPase will come through here, and we'll see a process just now. And in the process, it'll make ATP. Now let's watch this.
I'm sorry, Doc. Are we supposed to be hearing something?
Yeah, I'm sorry. It might not come through. It's my laptop thing. So anyway, I think I'll skip that one. It's really basically, it's just basically the explanation how ATPase works. Okay, but it's not that important. Okay, it's just nice to see what happens on a cellular level. It's actually mind-boggling. But anyway, don't worry about the voice so much. I think it has to do with the video, that the video voice doesn't come through on Zoom. Does that answer your question?
Noted, thank you.
Okay, so let's have a look. On the one end, we put glucose in the cellular cytoplasm, and it gets fermented and it makes 2 ATPs. Then it goes in the Krebs cycle and it's also fermented and it makes, you can use glutamine and it also makes 2 ATPs. And then the stuff that is important, that makes over or nearly 90% of the energy, is the oxidative phosphorylation. In other words, you take the protons and you push them through a certain protein that turns like a pump, and it creates ATP in the process. And as that happens, the protons are used up. This is if the mitochondria work very well, and you can see nearly 90%, 36 of ATP gets made for actually 1 molecule of glucose. So please remember this slide. It's an important one. It means that fermentation doesn't make a lot of energy. Fermentation either from glucose or either from glutamate. So what do mitochondria do at the end of the day? They make energy. They produce reactive oxygen species, which are molecules that have got too few electrons. You don't need to know the detail. We'll give you a summary just now. They're involved in signaling with calcium. They've also involved in signaling. It's called the reverse retrograde response. They also switch the body or the cells to kill themselves. It's involved in iron metabolism and hormone metabolisms, and it regulates your body temperature. So it's a lot of stuff they do. And this is a summary. Once again, these little chromosome pieces that are inside the matrix, this is a summary of it. It's only got 24 genes, 2 for RNAs, and so on. And this whole thing is what makes about 12 proteins or 14 proteins in the mitochondria. So let's look at this. If you say the mitochondria in total need 1,014 proteins, 1,000 are made from the nucleus of the cell, and only 14 are made in the mitochondria. Once these proteins are made in the cell, they have to be shipped into the proteins to work there. So you need a transport system. It's like building a car, and some car parts come from Italy, and some parts come from Germany, and some parts come from England, and eventually the car is put together in Belgium or wherever. And so you need parts from the nucleus, from the cell, and you need parts that are built in the factory itself, and you put it together. Okay, so the essential part here is we need proper respiration. In other words, oxygen and phosphate molecules to get combined to make ATP. We also need healthy mitochondrial DNA. This means the membrane potential. In other words, the electricity across the mitochondrial membrane has to be around plus minus 170 millivolts. Also, we need iron sulfur clusters. Those are specific enzymes. If they're not there, then the transport of the proteins into the mitochondria can't work. Then the mitochondria can't be assembled properly, and if they're not assembled properly, they cannot function properly. They are incredibly important in the feedback system, and then mitochondria need enough energy to have the cell's DNA repair mechanism. So we'll talk about that later again. These are all the different jobs that they do. They produce macromolecules, they send signal, they make energy, They tell the cells to kill themselves. Mitochondrial DNA, we've said, and then we've got reactive oxygen stress. So when things go wrong, here's the first example. So when the oxidative phosphorylation doesn't work, you've got too little oxygen, you've got mitochondrial DNA are damaged, the proteins don't work anymore, and you get this whole system going crazy. So the stress causes damage, the signals change, and the mitochondria will activate certain nuclear transcription factors. What are those? Those are like the little scripts that the waiter sends to the kitchen, and in the kitchen they start making a specific dish for you. Now, if there's a stress situation at the table, the waiter will send a note to the kitchen and say, listen, there's a problem, the customer is upset, the steak's not done well or whatever. So these are the transcription factors. And then certain genes will be activated and different proteins will be made. The second example is if mitochondrial DNA gets damaged by ethium bromide, which is a toxin, the oxphos is getting down, the membrane potential doesn't work anymore. The protein transport, the transport proteins from this side, from the cytosol to the mitochondria doesn't work. The electron transfer chain, which is really the generator, does not work anymore. And then the DNA repair mechanisms in the cell also can't work properly. So can you see where we're going? Outside damage damages oxphos inside mitochondria. All these things happen. The ETC doesn't work properly. And what do we see here? DNA damage starts accumulating because it doesn't get fixed. It's like a road where the government doesn't fix the potholes anymore. What will happen to that road? After 5 years, there will be no road anymore. So the people will start driving next to the road. You could say driving on the tar road was oxidative phosphorylation. You drive quickly, you drive efficiently, you save fuel. Now there's no road, you drive next to the road. Now you have to go to fermentation. You only get a little done, you drive slowly, and they don't work that well anymore. The next example. So here we have mitochondria. We've got dysbiosis, we've got lipopolysaccharides, and then we've got this guy, which comes from SARS-CoV-2, S1 spike protein. They act on all these factors, and they increase mTOR and they increase NF-kappa B, which are transcription factors. So mitochondria will get hotter and hotter and hotter, and they get more and more inflammation. And these Treg cells go down, and T helper 17 cells up inflammation, and eventually we get abnormal growth here on the right. Putting it differently, in a wound, when you have a wound, NF-kappa B is increased, cytokines are secreted, we get inflammation, the cells grow, apoptosis gets shut down, and you get tissue healing, tissue growth. Now, this is a good thing for a short time, but if it carries on growing and growing and growing, then the wound healing actually becomes like cancer. The fourth example is a retrograde response. So when the mitochondria are under stress, when things are not working well, they send a message to the nucleus and say, listen, guys, there's a problem. Things are not working. Things are not going the way it should. You have to switch to emergencies. In Southern Africa, we would call it load shedding. So each one gets a little bit of energy at specific times. And this is what they do. So when there's no oxygen, when there are lots of toxins, lots of reactive oxygen, mitochondrial DNA is damaged. The mitochondria say there's a problem, or membrane potential goes down. There's very little ATP. Calcium gets left in the cytosol, and then the cell switches. These, you could say, basically enzymes, and it activates new transcription factors in the cell. And what are these? Once again, NF-kappa B, they are all pro-inflammation. They make more and more and more inflammation. And inflammation never stops. Inflammation and growth, if it never stops, we've got a wound that keeps on growing and growing and growing. And that becomes like a tumor. So, and eventually that tumor tissue starts invading the tissue around it. What was interesting, the retrograde response can be switched off if you remove the initial triggers, at least in the, you could say, in the experimental environment. So the important part is that tumors take up glucose and they can ferment glutamine. They can also take stuff from other cells next to them that have died. And they use glycolysis for making ATP. They need nitrogen, and that they get from other dead cells. It's like vultures and jackal that eat from a carcass of a buffalo. And then there's gene activation by specific metabolites that come from cells and dead cells around the tumor cells. So there's a change in the microenvironment, and that, as we will see in future lectures, is a massive therapeutic opportunity. In other words, you can utilize it for yourself to actually attack cancer cells. So here we have a cancer. There was a primary cancer, I suppose maybe in the prostate here or in the bladder, and these are metastases that go to other body parts. This is a PET scan. Now, a PET scan is basically glucose molecules labeled with a radioactive substance. They get mixed and they get injected into the vein, and they go throughout the body, and they go specifically to the cancer tissues, because cancer tissues love glucose. They take them up very, very quickly. And this is why you can use high dose vitamin C and inject it into a person, and it'll go to cancer cells very rapidly, because vitamin C and glucose look very, very much alike. They are just about the same molecule. So this technique of making visible cancer cells is actually based on the fact that cancer cells love glucose. So when a tumor that has been sitting in one place for many, many years, and remember, everyone that is sitting and listening to me now has cancer. It's just that many of us, the cancer hasn't grown, and our immune system is keeping it in a small contained area. Once it's not contained anymore, it becomes into a tumor. Vessels grow in, it stops listening to each other, and it just starts growing. And then it goes into the tissues around it. Eventually, it'll go into the lymph system and into the blood system. It's called intravasation. Inside there, normally, macrophages and monocytes would have killed it. But now the cancer cells manage immune survival. They do something to the immune cells and say, listen, I'm not your enemy, I'm your friend. It's much like a policeman that's corrupt. He cooperates with the culprits, with the criminals. And now the police can't do its work anymore. And they can't be caught out by the immune system. So they circulate in the body, and they suppress certain areas of the immune system, and eventually they climb out of the vessel and they move back into another tissue. And often these tissues, specifically the lung, the liver, sometimes the bone, and sometimes even the brain, are tissues that have got very fine capillary beds. So they end up getting stuck in these fine capillary beds. They leave the vessel and they move into the tissue, and then they form secondary tumors. This is what it looks like graphically. So you have normal tissue and there's a tumor growing. It grows bigger and bigger and bigger. Eventually, it'll go through the little vessels into the big vessels, and it invades and it moves around the body. The immune system is sort of, you could say, compromised or corrupted. And eventually it ends up in the lung, and then it goes out of the blood vessels in the lung and it moves into the lung tissue. And ultimately you have got secondary tumors, metastasis in the lung. So what have metastasis got to do with this? The mitochondria, here's an example of a mitochondrium. Here is the cell wall of the cell, a double lipid membrane. These are little cell receptors. And this is just a short little summary of everything that the mitochondria do. It's just incredible. And it's like a factory with robots building cars. And I've said always, the invisible controls the visible. So the computerized mechanisms in this factory control the robots, and eventually they make a car. It's very much like mitochondria. So the cancer cycle is, you have got more electrons. This fifth complex is damaged, or 1 to 5 is damaged. The electron transfer chain is damaged. You have less protons, and if you've got less protons, less ATP gets made. But you've got lots of electrons and lots of oxygen around that doesn't get used. And those two things together make H2O, which is hydrogen peroxide, which is very toxic. And this is then switching on these specific transcription factors. And many different genes are changed, and then the cell becomes a cancer cell. And it has an altered metabolism. And here you see the result of it. So if this is all going a bit fast, at the end of today, I will provide Jackie or the Intellectus group with a PDF of this, which you guys can get from them and then look at this in peace and quiet and try and rebuild what I've been rushing through now. So once again, the retrograde response can be switched back by NAC and CoQ10. And by the way, CoQ10 is one of the most important enzymes in the electron transfer chains. For instance, if you give statins to a patient, it blocks the manufacture of cholesterol, but it also blocks the manufacture of CoQ10. So long story short, if you give a patient statins, you will damage his mitochondria and you will damage his electron transfer chain, thereby limiting the energy that is available from the mitochondria. Once again, another picture of what mitochondria do. You will also get this in your summary. If you leave here today, remember the mitochondria are actually two things. They sit inside our cell. There are many of them, sometimes 20,000. They are there part of the other organelles. This is the nucleus. And the mitochondria are like the little Honda generator that you need in Southern Africa, or South Africa, if there's load shedding. And also, they are like the central processor in your computer. So if you can take this picture home with you today, it'll be great, because then that is the fundamental message that I will give you today. So Thomas Seyfried said when we started, we need another model to explain cancer. I've given you a model. Why do small children get cancer? Because very likely something damaged the mitochondria in their mother, and the mother mitochondria are damaged already. They are given to the child, and the child gets another couple of damaging things that can happen to his body. And from there, something goes haywire in this little guy, especially in a modern environment. It might be chemicals, it might be food, it might be vaccinations with mercury or thimerosal, whatever in it, or aluminum. And you can understand now why every tumor is different, because the genetic damage is the downstream. It's the result of the upstream mitochondrial malfunction leading to a lack of DNA repair. So in all different places, the genes of the cell are damaged, and it doesn't happen to only one cell, it happens to every single cell. So the end result will be different in every single cell. And this is the explanation for that. Why are we stuck in investing so much time and effort in looking at genetic solutions? Because it's not a genetic problem. So if you put your ladder up the wrong wall and you try harder and harder do this thing and more efficiently, you shouldn't be doing at all that thing. You should be looking at other avenues to actually understand cancer and maybe use this model that is highly researched, and there's lots of work done by the Seyfried group that will prove that this thing is a very, very sound model. And so if we come back to the beginning, cancer is not a genetic aberration. Cancer is a mitochondrial aberration. So this is closing it up. Sorry, I must jump back one slide. Please understand that mitochondrial damage, and it can be from Wi-Fi routers, it can be from getting vaccinations, it can be from drugs that you give your patients, it can be from foods that have got probably different stuff, from viruses, from certain bacteria, many different things. They will damage the energy and the signaling, and you will end up with chronic disease. So if we want to tackle chronic disease, we have to understand that we have to look at mitochondrial medicine, as it is called. In other words, remove the triggers, rebuild your mitochondria, protect them, and do anything that you can to virtually reinvigorate your mitochondria. Ladies and gentlemen, thanks very much. I hope you enjoyed it. As I say, if you feel a bit overwhelmed, don't worry. We will give you the slides in a PDF format, and you can look at it in your own peace and quiet at home.
Thank you very much, Martin, for again challenging the paradigms and making us think. And if anybody is intimidated by this set of slides, I've got another 247 slides that Martin put together that that just make us realize that your biochemical knowledge is beyond belief. So I'd just like to thank you, and thank you for making it easier with the analogies of generators and computers. So I think there may be quite a few questions. There are a lot of comments. This is a perfect presentation. Informative, interesting, and easy to understand. Thank you. Well articulated, and the biology simplified. So thank you, Martin, for that.
It's a pleasure.
I'd be interested. I'm just looking for questions in the chat at the moment. I am aware that we are talking to Namibians, and we have hit our hourly allocation. So I just want to find out. I know Dr. Stephen Schmidt is on the call. I just wonder if he wants to make any comments.
Thank you, Jackie. Yeah, brilliant, Martin. Thanks very much. Unfortunately, the detail and the incredible insight that you've just presented is absent in the majority of practitioners' minds, and that is a sad thing. I just want to mention the interesting thing is, if you look at the history, and that is that Otto Warburg in the war, he was quite an interesting character. He was a Jewish, apparently gay guy who survived the war, and Hitler protected him because of his knowledge of cancer metabolism. He protected him during the war because Hitler was obsessed with cancer because his mother had breast cancer. And Warburg's theory would have saved humanity after that, but for the discovery of the human genome by Watson and Crick. So it's almost like it was hijacked. The whole idea was hijacked. Second thing I just want to say to everyone on this call and everyone who are treating people, you need to identify metabolic instability in the young. And it's getting younger and younger and younger. And it's very simple. Few basic blood tests, a little bit of assessment of blood pressure and weight, and you can identify metabolic instability, which is a collection of these metabolic or these triggers that Martin was talking about that eventually leads to this highly complicated change metabolism and cancers. So we need to identify these things in young people and change it, because you can do it simply. Certain things we cannot avoid. We can't avoid pollution. We cannot almost not avoid things like radiation, but we can certainly change our lifestyles, and any person can diminish his risk for malignancy and other chronic diseases, because they all basically work in the same way quite easily. Thanks, Martin. That was great.
Yes, and I'd like to reiterate, Doreen, brilliant presentation. One question has come up: why are the genes of breast cancer carried? And why do they cause breast cancer? Or is it both? Or is there a multitude of factors when it comes to the genes behind breast cancer?
I'm not a cancer specialist. I'm a biologist, so I look at it from a biology point of view. And I think Steve mentioned something there that I think is very, very fundamental to today's cancer industry, if I can call it like that. And that is our focus on genetics. Now, you can go absolutely bananas and crazy with looking at your genetics, and it's academically interesting. But for me, as a person who has to help other people and patients and give them sound advice, once I have their genetics, I say, okay, and now what? And luckily for us, genetics is one thing. But in most of the chronic diseases, the genetics only impart on, or only, you could say, between 75 to 90% is not genetic. Okay. So only 5% of genetics gives you a very, very specific outcome. And very often it's in very young patients already. In other words, you will see it early in life. A patient that has made 20 years and lived normally for 20 years, these genes are a predisposing factor. They make you more vulnerable. Like a person will have a slight build, a person will have a big build, you have a fair skin, you have a dark skin, whatever. And there are many differences in our setup that make us more or less vulnerable to certain symptoms and diseases. And I think the genetic setup in breast cancer is much that way. If you had these markers and you didn't have the other underlying basic mechanisms, as I try to illustrate now, I'm sure you would still be fine. And this is one of the issues that is so predominant. If you go to our traditional and very untouched communities in our countries, you find very, very little cancer, okay? And you find very little of these chronic diseases. And this is the fascinating thing. And we in Africa are in a unique situation to open our eyes and open our ears and learn from that. It doesn't mean we have to live like these poor communities, or go back to just cooking with wood. But we have to take the lessons from it and try and teach our children. In fact, many of my younger patients that come in, you would say their grandfather and grandmother lived in Namibia in the north. Some of them are 90, some are up to 100 years old, and they die a peaceful death within their communities, within their families. They age gracefully. They are part of the family. They're not in a hospital. They don't lie in a hospice with cancer for 10 years. They don't have diabetes and lose their limbs and their eyesight and all that sort of stuff. And this is something that we can learn from and try and ask questions. And the big thing is, when you want to have an answer for something that you observe, ask questions. Ask questions time and time again. I will just put one trigger into this conversation tonight. The spike protein from the coronavirus, number 1, and number 2 from the vaccine, is possibly the most toxic protein that we know for mitochondria. So long COVID and everything that goes with it has as its central pivoting point mitochondrial damage and dysfunction. And then you can understand what comes from it. Mitochondria are the predominant switchboard for our immune system. If they are damaged, our immune system doesn't work. And what can you expect? Turbo cancers, okay, for instance. It's actually all been shown already. The data are out, they're available. And so we can, once again, we can learn from that, and we can try and improve our understanding.
Can I just add to that? Can I just add to that? Just a very simple way of looking at that is that genotype is not phenotype. You have the genes, you don't have to have the disease. And there are very few classic examples of, there was one specific, I think it was Japanese or Korean twin sisters. One remained in a country of origin and birth, and the other one went to the Americas. And they carried the same genes. The one who remained and ate traditionally did not get breast cancer, the other one did. That is an example of one. The other thing is, if you look at the curve of distribution of breast cancer in a massive French study that was done, if you look at like a bell curve, 5% with true genetic disease, 5 or 10% with age, and the big bell in the middle was identified as being closely related to metabolic instability, meaning eating wrong, not exercising, radiation, pesticides, smoking, and all the things that causes insulin resistance. Cancer of the breast is not primarily a genetic disease. There are too many gene mutations. It is mitochondrial damage, as Martin so correctly said.
There's another interesting observation. If you go to popular TV channels, like the channels that are predominantly provided for the mass of people in our communities, from say DSTV or these type of rental guys, the advertisements and the human actions that are in these adverts, you could virtually look at every single one of them and say, this is one of the drivers of metabolic disease, of metabolic damage. And by the way, sugar also damages mitochondria, because mitochondria are like a petrol engine that gets fuel in one end and oxygen in the other, and it makes energy, and it has exhaust tubes. The exhaust tubes are the reactive oxygen species. The energy is the power on the wheel, the ATP. But if you push more and more and more fuel into the carburetor, the engine will drown. It will stop. It'll stop running. First, it'll stop smoking black, and then it'll stop. And this is what mitochondria do if you put too much sugar in it. So we all know these normal processed junk foods that are advertised, the cell phones on kiddies' laps, the cell phones next to a child's ear, they are damaging the structures adjacent to it. So if you look at a normal modern TV program with a family sitting there, each one has a laptop in their lap, one's got an earphone in the ear, and then especially these Bluetooth earplugs that are next to your brain. And it's a modern phenomenon. Everybody thinks it's cool. Those things have got incredible damaging effects on the surrounding tissues.
Jackie, can I make another comment? Can I make another comment about?
Yeah, no, you guys go with the panel discussion. I just want to say to the audience that I mean, most of them are still on. So if you want to leave, complete the form and leave. But I'm fascinated with this discussion, so I'm happy to continue with it.
As far as the genetics go, one must understand. Go to history again. If you look at the statistics, official statistics of the cause of death in the 1910, the top 10 causes or 8 causes of death were all infectious. All the infections of the various organs. 100 years later, the top causes of death was cardiovascular disease and cancer. And the infections went away because of mostly antibiotics, sanitation, and. But you cannot change, the genome cannot change that radically in 100 years. That's just an endorsement of the whole concept of it is not a somatic problem, it's a metabolic problem. It is not a genetic disease. It is an epigenetic disease. In other words, your genes are ruined by the things that Martin so clearly stated. And with regards to sugar, just want to expand on that a little bit. What we eat today as sugar, as sweetness, I didn't eat as a child, because you are getting fructose, high fructose corn syrup in all your sweetness, and fructose is a really damaging molecule. And together with that, the seed oils. Our grandparents, and when I was a child, our food was cooked in animal fats. It's now cooked in seed oils, which are highly toxic. Again, the mitochondria suffers.
Look, it's actually easy. I think all of us here that live in Africa, we have got prime examples of rural communities. When you go to rural communities and you drive past these schools, what do you see? You see youngsters, school-going children, teenagers. In the morning, you will see them walking for 5, sometimes even longer, to get to school. So that's the normal morning exercise. They do it playfully. They don't even think it's exercise. The second you see when you drive past the school, there are very few children with cell phones sitting in a corner. They're dancing around, they're running around, they're dancing and singing and smiling and chasing each other. It's a fascinating phenomenon. You don't see fat people. You only see fat people within 50 kilometers of a modernized settlement with modern foods, service stations, all the stuff that you buy in the quick shop at the service station. And because these people work there, they're often the first ones to succumb to this. Then once these people move to the big towns, they start eating, and especially if they earn a good salary, they start going to restaurants, fast food restaurants. They go to restaurants like Steers, and where the traditional American diet is served. And number one, the parents will get fat. Number two, the children will have all the issues that modern children have. They spend their time with mobile devices. They don't exercise anymore. And guess what? In my field, in dentistry, within 10 years, we make it 2 generations. Within 1 generation, you see dental malformations. All of a sudden, they need orthodontics, which you will never see in rural communities. In fact, rural communities in Africa, I would guess 80 to 90% have got 32 teeth. They've got virtually no caries until they get to modern food. And this is not just in Africa. It's been shown worldwide. It's nothing new.
No, I'd agree with that. And thank you for saying it. It's been an incredibly interesting discussion. I know it's the first of 4 series. There are a couple of other questions in the link. Just to introduce everybody, Dr. Stephen Schmidt is a very well-known gastroenterologist in South Africa and a hepatologist. So I didn't introduce you, Stephen, because I know there were issues that you might not be able to get on the call. So we're delighted that you're here. And one of the questions that probably Stephen could answer better than Martin is on the question of intravenous vitamin C. Can injecting or can high-dose IV vitamin C help with cancer?
Yeah, you actually should ask Martin. Look, the vitamin C, the first part of vitamin C, Martin already mentioned. There's similarities between glucose and vitamin C. Vitamin C is an interesting molecule in the sense that if you take vitamin C by mouth, it has a different effect than when you take it intravenously. Vitamin C can be an antioxidant and it can be an oxidant by itself. So there are lots of theories about exactly how this works, but the major part of how vitamin C works in the management of cancer would be as an oxidant. But here is something that I can say as a gastroenterologist, and that is that vitamin C is essential to the maintenance of a healthy microbiome. Now, that's a whole dog of its own, but the microbiome or the microorganisms and its massive genetic substance in the gut is massively controlling to chronic disease. And that is a whole new and very new, exciting field. And when you take vitamin C by mouth, about half of it feeds the microbiome and can actually fix your microbiome, and the rest of it would have its good effect. Martin, maybe you can just add something to the IV vitamin C.
Yeah, look, vitamin C per os, I think in most people, when you get to about 3 or 4 or even 5 gram, your body doesn't absorb it anymore. In fact, you get runny tummies and you actually
Your body doesn't smoke.
There is a technique, and it's called C flush, that if you want to get your bowel cleansed out in a reasonably safe and effective way, you just take some vitamin C, you can stir it in powder form into water, and you start drinking it bit by bit by bit until you get a runny tummy, and then you end up going to the toilet within the next hour or two, and everything gets flushed out. So there is a limit on how much vitamin C you get into the system. And when you get the oral vitamin C, and it's an antioxidant. Antioxidants are molecules that have got extra electrons. They are electron donors. They can give electrons. So free radicals in the body are like the black fumes that come from a car exhaust. And those are molecules that have got too few electrons. So now you take the antioxidant, the electron donors in vitamin C and vitamin D and whatever you use that give the electrons to the free radicals, and they neutralize them, and therefore the body can handle them. So it's different when you take vitamin C and you start infusing it. You start with about 7.5 grams, and you can go up to crazy amounts, up to 220 grams over 24 hours. It's not to be done for a guy that hasn't been trained, and I don't do it because I'm a dentist, so I don't want to go that way. It works incredibly well in certain situations. The body uses massive of vitamin C, and you can only get those amounts of vitamin C into the body via veins. And then, as Stephen says, the antioxidant action of vitamin C turns around, and it becomes a pro-oxidant. It becomes stressful. And one of my lectures next year will be on cancer support programs. I don't want to call them treatment, I call them cancer support programs. My situation at the moment is that people have got cancer, they come to us and they say, what can I do? And I say, go and speak to your oncologist, do whatever he wants, finish what he suggests, and then when you're done and dusted, when the acute phase is over, we will come and we will provide you with the logic and the research and a program that will help your body manage, on the one hand, the cancer, but mostly after the treatment, it's the downstream damage that the modern cancer treatments leave in your body. So in that, we use metabolic therapy, limiting sugar, limiting glucose, using ketogenic diets, and then also giving certain triggers that cancer cells don't like. And one of them is high dose vitamin C, or on the other hand, you can use hyperbaric oxygen and others. And hyperbaric oxygen does the same. It's a pro-oxidant trigger in the body. And if the cancer cells are weakened by having too little glucose, and you club them and you nail them with a pro-oxidant therapy, you kill a lot of them. But you have to do it in cycles, and you have to do it in a very specific way. But that is part and parcel of a future lecture.
Great. Thank you for that, Martin. I am very aware of time. I'm also aware that some of the staff recording are going to need to leave. So can we end the recording there, please, Pride?